What's up, everybody? It's MycoGeeky here with MycoGeeky Podcast, the podcast that goes deep so you can level up your at-home mycology game. Tonight, we're going to get down to business. We had a technical difficulty last week. Looks like we might have a technical difficulty this week, but I'm going to kill a little time. We're going to see how it goes. Hold on one second. Just got to message here from Alan. I'm having internet trouble. One second, I'm going to message him. Okay. Here we go. All right, sorry about that. That definitely was not how I planned to start tonight. So here we are yet again, Monday night, 9 p.m. Hopefully you guys are ready to do some learning, because whether Alan shows up or not, we're definitely going to be doing some going deep and learning a few things. I do want to give you guys a heads up. For 2023, there's going to be some format changes. I'm going to try to produce the podcast a little bit more and have it more structured and segmented. So there will be different sections. You know, there will be a grower spotlight. There will be a tech section. There will be a genetics section. There will be really deep in the science section, all sorts of things, just to try to broaden the flavor for everybody, so everybody's got a little something they can get into. That way, if you don't want to talk about grain for 3 hours, you don't have to listen to me talk about grain for 3 hours. We'll do other things anyway. So just want to give you guys a heads up. We got a lot of great podcasts coming up next week. I got my buddy Slightly Feral. He is an extremely unorthodox grower. He always manages to surprise me with the things he's trying and the things that are working. So he's going to be pretty great. And he's been growing a heck of a long time, and he's grown everything. If it's a mushroom, he's probably tried to grow it. So look forward to talking with him next week. And then in the following week, we have the one, the only Jake Onsid. He is going to be talking to us about growing some Pancyans, if you know what I'm talking about. And hopefully we're going to do a few Pansian podcasts here, and everybody can maybe dip their toes in that dirty yet luscious Pansian water, aka Substrate. Anyway, so then after Jake, we're going to have a special podcast that's going to highlight some female mycologists. I don't know if you guys have been paying attention, but female mycologists are killing it. And we're going to talk about that. And we're going to talk about what it's like being a female in an extremely male-dominated community. I think on my analytics, I think it's like 98% of my viewers are guys. So, it's a small fraction of my viewership are actually female. And yet, if you start looking at some of the work that at-home mycologists are doing, there's a lot of females doing some really strong work. So that's going to be a fun one as well. And then we got Wumbo Myco coming back. He's got a new innovation he wants to talk about, and we're going to, we'll talk about other things, but we're going to have him back. It's going to be quite fun. Anyway, let me get a little update here. There is no update. Okay. I know you guys think I'm doing this on purpose. Like this is like the world's greatest suspense build up to Alan Rockefeller. I promise you, I cannot take credit for that. It's all his publicist, guys. He said, I have an idea. We're going to do this he doesn't show up thing for a while so that the anticipation builds and builds. No, I'm kidding. It's not. He's just a busy guy. He right now is having some internet troubles. But so last week, our citizen scientist, ex-college professor, ex-mycologist from Tennessee, now in Thailand, Ed Grand, he is going to hopefully, let me see if we got him ready. We do. I'm going to pull him back up. Ed and I are actually planning a lot of things in the future, a lot of educational videos. So for those of you who take science seriously and like to grow mushrooms and want to merge those two a little bit better, we're going to have a lot of really in-depth content for you guys coming up. So I'm going to bring him on, and we're going to get into a little bit of that stuff right now. So without further ado, welcome back, Ed Grand, Ed 1K. What's up, dude?
Hi, you guys.
Hey, man.
This is out okay.
Yep, you can turn your mic up. Sorry, guys, we didn't get a sound check or a tech meeting beforehand here, so let us work this out a little bit.
Oh yeah.
Just turn that volume up. Look
Okay,
at Ed. It's 9 a.m. over in Thailand. He's freshly shaven and ready to go. He might even have pants on this week, guys.
I do actually have pants. I still got the socks though. I actually went off to find some really nice headphones, but there was so many of them. Wow, the gaming headphones. Oh my gosh.
If you think there's a lot of gaming headphones, wait till you go shopping for gaming chairs.
Yeah. Oh my God. I saw a chair that cost like $1,000. I was like, really? I thought they put one too many zeros on it. I was like, oh my gosh, really? I was like looking at it, like, did they write the right?
I mean, well, those are modern marvels. Those chairs allow a young 15-year-old boy to sit in them for, weeks at a time. I think there's little levers you can pull. You can actually just take dumps in the chair, and you don't ever have to leave the chair. It's pretty great.
I can imagine. It reminds me of the Idiocracy movie where he was going to the bathroom in the chair and all that kind of stuff.
Yeah. Can you turn your mic up a little bit more?
Yeah.
You might need to go to settings and see where your level's at and see if that's maxed out. If not, then it'll be on your computer.
Yeah, I had this problem a few weeks ago.
See, guys, it seems like I just show up and we just start talking, and we do that, but there's a little bit of preemptive work that goes into this. So I really thought this one was going to go off without a hitch this week. So my apologies. All right. My buddy Rounder saying Ed sounds great.
Does it sound okay now?
Oh, that sounds good to me. We'll get some confirmation. Everybody in the chat, let me know how the two of us are sounding. Oh, great. Perfect. People are saying he sounds good. Okay, all right. It's just on my end. I got this weird double monitor thing. That might be what's going on.
Yeah.
It is. It's me. It's me. My bad, dude. Sorry, guys. I didn't have one.
Okay, good. Because I didn't know what I was going to do because I...
Yeah. All right, guys. Well, we are more than 5 minutes in. At some point in time, I might see Alan's feed pop up, in which case we'll try to make a nice segue. Although we'll see how it goes here. I did, in order to have a really nice feed, I did have to run an actual Ethernet cord way over there where my router is in a corner, and I had to run it all the way down here just so I could have that. So some people don't have that, and it makes things a little difficult, but we're all doing the best that we can. So anyway, let me try to get my bearings. Ed and I'll give you guys a heads up. Hold on. I left the book over here. I'm going to just grab it real fast.
Okay. Yeah, I don't have my copy. I might actually have a copy. Oh, if you got it. Yeah,
all right. Hold on. I should have had Ed sing you guys a traditional Thailand lullaby or something while I was doing that, but I didn't even think about it.
Sorry, I'm a little sleepy. I slept for like an hour and a half or so I'm still a little bit perking up.
Well, dude, maybe if you weren't crossing so many Netherlands with just about everything on fucking Earth, you'd get more sleep. I think Ed has literally done like 19 crosses in the last day. He's hiding all this, and then all of a sudden he just goes, "Look what I did! Look what I did! Look what I did! Look what I did!"
Yeah,
exactly. He's joining the Julian Matucci and Yoshi Amano club of just what are we going to cross? Maybe 20 things this weekend. Who knows? These guys must have the largest shelving units on Earth. Is all I can say.
I have to go to buy a new shelf today. I've been trying to scatter them around, but I'm going to have to go buy an actual shelf today. It'll be a $50 investment to save me a lot of headache. It's like I can't even walk through my living room anymore. Thank goodness I live alone.
The shelving is clutch. Dude, if I lived alone, there is no room in this house that would not have a tub in it. I can tell you that.
Oh, I know, it's ridiculous.
Yeah.
They multiply like those Star Trek thingies, dweebles. What did they call it? Furbies.
Oh yeah, I remember that episode.
Little things.
Yeah, it was also like those Sesame Street things that were like rot, 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 rot.
Yeah, I can't remember the name of either of them.
Yep. Yeah, those guys.
Tribbles.
Weebles, yeah.
Tribbles, trifles.
I don't know, man. One of you guys in the voice chat's gonna have the answer for us in no time, I'm sure.
Yeah.
Anyway, so as Ed and I have gotten to know each other, he we just talk about mycology probably way too much, and he's always telling me, hey, you should read about this, read about that, read about the other thing. And so I've been doing a little bit of that. And he was saying that, it might be great if we're all sitting around here calling ourselves at-home mycologists. I think one day he said something to the effect of, I mean, it's a fair statement to call yourself a mycologist if you're using current scientific practices and methodologies to study fungi and mushrooms like that. That's pretty much all you really have to be doing. You don't have to have a PhD. You don't have to have a master's. You don't even have to have a bachelor's in science. You just have to want to know the things that those people who have those pedigrees tend to know, which I'm sure he will concur. There are plenty of people in this community that can teach any PhD mycologist a thing or two, particularly as it pertains to cultivation practices. I was just in prepping for Alan. I was reading up on one of the papers he co-published, and they're literally referencing a shroomery post in scientific literature, which is kind of hilarious. Anyway, so Ed and I were talking, saying it might be cool. People are foraging, and as people are more interested, especially in psilocybe and other actives, but just foraging in general seems to be a more popular activity. That it might be great to start talking about ways people can broaden and deepen their ability to forage for and identify mushrooms in the wild. So we were talking about that, and he said, well, there's one book that's sort of the Bible of this. This is the starting point, and I just got it in the mail today. How to Identify Mushrooms to Genus 1. I think there's 5 or 6 of these books. Of course, Ed says they're not all page turners. Surprise, surprise. Said some are better than others, but the macroscopic features book is the first book, and it is a good book.
Definitely. It's gosh, I think it was published. I don't know. I think maybe the early 80s. It was right around the time Stamets was published in the Mushroom Cultivator.
Okay.
And who is it? Daniel Stuntz, I think, is the primary author on there. I don't have my copy.
This one says David Largent.
Oh, that's right.
It's French or it's Largent. I don't know. It says the key is by Daniel Stuntz. Yep.
Yeah, that's right. Yeah, the key is what's important. I guess that you want me to. The way I started out with identifying mushrooms was with what they call dichotomous keys. Okay. So if you open that book or most books within the first 20 pages, probably more like first 10 pages, there will be what's called a dichotomous key. And a dichotomous key is going to ask you things like primarily
this or that, this, right?
Yeah, exactly.
Does it have this or does it have that? Is it tall? Is it short? Does it smell like this?
Yeah, exactly. Okay. And preferably to make you from going insane, it'll ask you one question at a time. So sometimes, even though they sometimes just call them keys, it'll often say, like, for instance, a lot of the first probably question it'll ask you is, what is the spore color? So in a mushroom that you would find out in the woods, you could often look on the underside and the gills, and you could see the spore color. Sometimes you might actually have to make a spore print, which would typically be done on a white sheet of paper, or sometimes they used to sell at some forays, they would have these Post-it note size of paper, which were half black and half white. And so when you put a mushroom cap on there, you could see if it had white spores, they're not going to show up very well on the white side, but they'll be on the black side. And you can also see the contrast. So for take an example, I don't know, like a lactarius, like a milk mushroom. Let's see. Or a lot of our mushrooms are white spored. So a lot of the edible mushrooms like the shiitakes, the oyster mushrooms, the flammulina, the enoki, they'll have white spores. So that's just kind of a really basic feature. Of course, our psilocybes would have a dark or a purple brown is the kind of traditional color, but we know that's not always the case, right? So those are ways to just get you sort of down the right path to basically the genus. So that book that you just showed, that's going to show you hopefully the right path to the genus of the
so the thing you're talking about, so in the end there's a lot of all the genuses, all the generic descriptions of all the genuses. Starts out with Agaricus, but then in the beginning, see, it might have been like 20 years since you read this book, Ed. You just memorized the important parts.
I have a lot of that book memorized. Yes.
Yeah, I'm sure you do. So introduction, what's a freaking mushroom, how to use this book, recognizing features, correlating measurements of the fruiting body, color, shape. Oh, here you go. Shape of the spore-bearing layer, characteristics of the pilus, the size, the shape. In my prep of getting ready to talk to Alan about some of his published literature, I learned adjectives and adverbs I had never seen before and realized I really didn't know much when it came to identifying mushrooms. I like them.
Yeah.
I haven't learned all the words for them yet, though.
A lot of it is going to be terminology that most normal people are not familiar with. It's going to be basically so that book and that group of people were specifically interested in what we call agarics. So they would be the guild mushrooms. But if you had something like if people know maitake or hen of the woods or chicken of the woods, that mushroom, if you know, it has pores on the bottom.
Polypore type.
So that would be an obvious way to separate what we would call the polypores, which used to all be in the genus Polyporus or Polyporus. That would be a fairly easy way to separate the guild mushrooms or the agarics from the polypores. So, well, then you got another small problem is because there's other mushrooms that have pores, the boletes. So things like Seps or Porcini or Steinpilz. I was talking to a German guy last night, Steinpilz. The ones that some people call the king boletes, I guess, out west in the U.S. They're called king boletes.
The ones everybody likes to just do videos of themselves tapping on, basically.
Yeah, they look like a little hamburger with a big giant. They're quite attractive mushrooms, and they can be quite delicious. Unfortunately, a lot of times they're full of like maggot and fly larvae, but
yum.
Yeah, Michigan, we out west, I don't know, it could be a slightly different. It's a species complex of Boletus edulis. And I'm not even sure, it might be in Boletus. They've separated that into many, many different genera now, but everybody would know Boletus edulis. And it's maybe a species complex that has a lot of different what we call cryptic species sometimes. So there's some terms. Yeah, cryptic, as the name kind of implies, they're like hidden species. So when you have a species like Boletus edulis, say on the west coast and the east coast, it's the same thing with chanterelles, like in Europe or America, they look very, very similar. But when you actually look at them in really close, like for instance, for chanterelles, I know the ones that grow out in California, way bigger. So what used to be called Cantharellus siberius, I believe now is about 6 different species in North America. There's a second problem in that Cantharellus siberius is a European name. So if you're European and you come to Michigan and you see a chanterelle, the typical orange chanterelle, which also grow here in Thailand, but they're tiny. Everything here in Asia is like tinier and cuter. And so they do.
I mean, they like it petite. And I had a Japanese friend who's like, "Oh, we don't like that big. No, big is bad. We like small." Yes. Okay.
Everything, even the mushrooms. Okay, we gotta be careful, man. Yes. So the cantaloupe
all
here
in
are a
good
little bit.
fun. All in good fun.
Oh, we good fun. Yeah. But yeah, if you go out west, and the first time I saw a chanterelle in California, I was like, oh my gosh, this thing is like huge.
Oh yeah.
One mushroom weighed like a half pound, and the little tiny ones you get in Michigan, Siberias, which is the typical like orange one. Smells like apricots, at least that's what people say. But there's other subtle differences like that. The chanterelles in Michigan don't really smell that much, but when you find them out west in Colorado or like California, they smell like that kind of apricot smell that you read in all the field guides. Mm-hmm. So those are things that you start to wonder.
It's like that Justin Bieber song. He gets his chanterelles out in California. Oh no, that's weed. Never mind. My bad. Sorry. Okay, keep going. Sorry, little pop culture reference there. I don't.
Too. Yeah. Okay. Some of those Humboldt like trees, that are like, that's a weed plant. Like really, it's like the size of a house. Yeah. So you start off with. So then again, if you I got a thing queued up here. If you just put if you just start going through these dichotomous keys, thank goodness some people have put them online and they're sort of digitized, so they have hyperlinks. Oh, cool. So in the first question, mushroom expert is one. I don't know how to comment in the chat here.
Yeah, you can share it though too. If you do, you want to show it?
No, I'll just type. I'll just put the URL. I think
okay, mushroom expert.
Do it in the old window.
Okay.
There we go. I think that'll work.
And we can add this again when I change over all the stuff. Okay, so for those of you who just joined, man, they're trying to keep us away from Alan Rockefeller. Yo, what's going on? I don't get it.
Get kidnapped.
First it was just the love of the game, just the love of that forage. And, getting lost track of time, being out in the middle of Eureka, California. California is not all Sunset Strip, guys. There is a lot of rocks and a lot of trees, and that's where he was. And then this week, didn't hear from him. We went live, and he just messaged me not too long ago saying, Comcast said there's a large outage. They would get it fixed by 5:30, but that was 45 minutes ago. So he just literally waiting to have internet access. Like I said, man, it's going to happen. This is like the world's greatest buildup. But in the meantime, for those of you just tuned in, we have Ed Grant, Ed 1K back, and we're going to talk tonight about a few things, depending upon if or when Alan shows up. But we were planning on getting into how to identify mushrooms to genus. The idea being anybody that takes at-home mycology seriously and wants to level up and wants to be able to go out with their friends and forage for mushrooms. If you want to actually identify the mushrooms that you're looking for and finding, you have to have a process for that. And Ed was just talking about dichotomous keys. This book, what was that website that you just mentioned?
Mushroomexpert.com.
Mushroomexpert.com is a place you can go, and it just starts asking you questions about the mushroom you're trying to identify. I believe I at one time used an iPhone app that did a similar thing. I think it used photos, and then it also asked questions, and so there's things like that exist. So anyway, so keep going. You were talking about chanterelles and how they look different in California versus Michigan versus Thailand. Okay, keep going now that we got everybody caught up.
Yeah, so once you get past the basic, so you're gonna have things like basically your guild mushrooms, which are the typical mushrooms we know, and then you're gonna have your polypores. And then when you get to groups like the chanterelles, they can be quite distinctive. So they'll often split those off early in the keys. So if you have a picture guide, that's really nice, even sort of like a cartoonish sort of drawing.
Right.
So they usually try to split, like puffballs is another one. Puffballs are pretty obvious to most people. And so you'll split off the puffballs early in the key. They'll basically say, like, does it look like a puffball? Does it look like a chanterelle? Does it look like a bolete? And then you get to the guild ones. And that's why that book that you showed was written. Because the guild fungi are the majority of things that you're going to find, probably that you're going to not know what they are. So everybody, once they see a giant puffball, knows what a giant puffball is. You don't need a book. You don't need a key. You don't need iNaturalist to tell you what a chicken of the woods or a giant puffball. There's some of the edible ones, especially that they're so obvious that even if you were blind, you'd probably be able to recognize them by their smell and their shape and their feel and their weight. But when you get to the guild mushrooms, that's when things get messy. And the primary thing to differentiate guild mushrooms is spore print color.
Okay.
It's a traditional character that still has relevance, very, very significant relevance, because it's very simple to do. It's very consistent. So if I find a lactarius, what I think is a lactarius, a milk mushroom, lactic acid, lactose, it's going to have white spores. It's going to have white spores. If it has dark spores, it's not a lactarius. There are no lactarius that have dark spores. So if you find something that is supposed to have light color, there are lactarius that have like tan or cream colored spores. Then you get into those color wheels, for the internet, designers, the color wheels and what's cream and what's beige and what's tan and what's milk and what's ivory.
So I'm reading here. This book says brighter yellow colored, including white, pale cream, cream colored, pale to bright yellow, greenish in one case, or then a salmon to pink, and then yellow to brown, and then purple brown to chocolate brown. That's the one I like. And then spores, smoke color to dark gray brown to black.
Yes.
I didn't know there were black spores. Those are like the fucking metal spores, man.
No, those are the Norwegian mushrooms. They all have black spores. Norwegian and the Finnish black metal spores.
Right.
No, Caprinus, shaggy manes. Shaggy manes would be the most common one. Yeah, so shaggy manes, you typically can't get a spore print because of Cap deliquescens. It like auto digests.
Right.
But if you did, a cool thing about shaggy manes, they used to make ink out of them. One of the, I forgot which war it was, but one of the sides was putting Caprinus spores in their inks as an indication that was the correct communication.
Oh, wow.
So they would receive these communiques or whatever, and they would look at the ink, and they had been putting shaggy mane inky cap mushroom spores in the ink. And
they could have just written at the bottom for microscopy purposes only, though, right?
But let's see. Caprinus is black spores. You got to be careful too. Some of our like lookalikes, if you're out in the woods looking for psilocybes and you have the unfortunate circumstances to run across a Galerina. Galerina is one of the deadly LBMs. Fortunately for us, they grow on wood, and like really they usually grow on like logs. So they'll be on the like underside of a log or somewhere in a little knot, So they're not going to grow on like wood chips. Probably you're not going to see them there. They're not like the thing you're going to find out in your mulch. They're going to be in the woods on a log. So they have kind of darker spores. Black though is really, I can't, Caprinus, I think, and Satharella and pancyans will have black spores. If you find a pancyan, it should have very, very, very dark spores, black spores. Yeah. So our spores are more for a wild cubensis. They're more on the kind of like purple brown, depending on how the light's hitting them. But Paniolus or Coplandia pancyans, they'll have black spores, like jet black spores.
Oh, okay.
So that is good because, again, if you're looking for something fun, you don't want to confuse it with another LBM. There are LBMs that'll have kind of brown spores. There's some random things that you're just, like little crappy things you don't really want to bother with. Okay. But pans, they should have jet black spores. So if you
now,
see a spore,
you know what this reminds me of? As we're getting into the dichotomous keys and some of these very, like the first steps, like just take a print, what color are the spores? Yeah. And then you can just eliminate like a whole ton of things by doing that. It reminds me of microbiology, where I'm doing a certain gram stain, and if it stains, then I can eliminate all this stuff. And so I'm imagining it's very similar where You just have these different questions, and the more questions you ask and you accurately answer, the closer you get to honing in on, well, like this book is saying, you're getting to genus. So you're just one step away from knowing exactly what the hell it is.
Yep, it's exactly the same. Okay. It's exactly the same with microbiome, with bacteria. They don't really have any morphological characteristics. They're too small. Most of them are. Yeah, most of them bacilli this big that are like 1 micron long, which is even for at 1000x, you don't see anything. And there's thousands of species of bacteria, and as you know, they're differentiated based on their biochemical properties primarily. So, like you said, the first thing, gram-negative, gram-positive, which is essentially a chemical test for certain types of components in the cell wall.
Right.
And then
is it aerobic, anaerobic? Right. Does it take a certain stain? I can't remember all the stains we used, but we used a few different stains.
Yeah. They're all basically ways to visualize a chemical reaction, which usually revolves around an enzyme that they produce. So peroxidase is the enzyme, ACE, that produce. So if you see ASE at the end, that means it's an enzyme. So peroxidase, if you take hydrogen peroxide, like the stuff you would clean a cut with, and you take a little dab of your bacteria and you drop it in there, it'll immediately start to bubble. So there it's, you go through this positive. So it's a dichotomous key, right? So it's either peroxidase positive or peroxidase negative, and then you move on to the next question. So the problem there for a lot of beginners is that you'll often come to a question like that one you mentioned. What is the difference between brown and olive brown?
Right.
And when you're looking at, say, I don't know, like a russula that has a green cap, well, is it like light green or dark green or like olive green or mint green? They actually have old color charts like you would see at a paint shop, where they have those strips of different colors of like orange, and you want to match it to your, your bathroom wall or whatever. They have those things. They have those books for mycologists and for artists, for paint manufacturers.
And so so is this why frequently in the articles I'm reading lately, they'll tell you what microscope they use. They try to give you as much like photographic information about how the photos were obtained. And I'm almost like, does it matter that it was a Zeiss such and such or a Nikon this or but I'm guessing it's to be able to potentially recreate and understand how whatever image you're seeing it was created, right? So that color accuracy is more accurate.
Yes, reproducibility. Yes, exactly.
So now I got another question for you. We're not doing any of this with our cultigens, Ed.
I know.
We're not doing any. I'm not doing it. None of us are doing it. All of us at home mycologists, we're not doing. I could not use this dichotomous key yet. I will be. Don't you worry, guys. To identify, a cultigen I'm working on. How do I describe a Melmac? How do I describe a Melmac Reaver F3? How do I describe Shakti Blue Brains? How do you know what I'm saying? Like,
yes,
it would up our game to be able to do this, right?
Okay. So it's kind of the same. I don't know. I just thought about this. Every time I talk to you, I think about new things.
Great.
So if you took a corn plant and you gave it to a Native American 200 years ago, they would have looked at you and be like, "What the fuck is this?"
Right.
Right? Like, what is this? Like, that corn that we put out on Halloween that's like purple and green. They might have had it. They might have seen it somewhere, but they wouldn't probably recognize, an 8-foot tall, Monsanto derived cultigen. That's
wait, Monsanto? Well, they're not they didn't just celebrate their 2000th anniversary. Oh, okay.
Oh, now they're Bayer. I think they're all Bayer now. Bayer has like absorbed all their legal.
Even better. I love it when drug companies buy agricultural companies. That'll never be a problem.
I know. I think it was like $60 billion. I think.
Oh my God. Well, I mean, the drug companies are making so much money, they got to buy something.
I know.
Bill Gates is buying all the farmland, so
exactly. There's like some some executive at Bayer was like, I'm bored. Let's go buy Monsanto today. Like,
It was between Arkansas and Monsanto, and they picked Monsanto.
They want to get in the cannabis industry, man. Yep. They see a lot of money there, and they want to get in on it early. Have GMO weed in a couple more years.
Oh my God! I'm surprised that doesn't already exist.
I bet it does in a lab somewhere underground in some salt mine in probably under Detroit.
All right. Hey, Dave Wombat, if you're still here and listening, we need the name of the non-GMO weed strain that Monsanto will come out with soon. Please bless us with your creativity on this one.
I saw Dave somewhere in the comments, one of the comments.
So, he was just making fun of you using big words. That's all.
Yeah. I'm sorry. Sometimes you got to use big words. I've been trying to keep it gotta PC or whatever, but sometimes I gotta be done.
So here's my initial thought, because yes, I would love to be the Myco cool kid, maybe not really the cool kid, and be able to go out and identify mushrooms in a more scientific way than how my uncle did it, which was he just knew every mushroom. And I don't think he really specifically knew how he knew it. He just knew it. But the scientific methodology is important. And in my head, and in 2023, I want to start talking more about code of ethics. How do we sustain ourselves as a community? How do we make best practices and kind of show that we deserve the limelight that we're being given? And part of that is, naming things. And is it really a new isolation? And what does an isolation even mean? And all these considerations, which, as I'm sitting here thinking about this Identifying Mushrooms to Genus book, I'm thinking, man, if I think I have an isolation of, let's say, I don't know, Casper, right? So I got Casper. I love it. I'm running it, but I think I did it different. Now I got Casper's brother, Basper. I don't know. How do I need to both be able to identify Casper and then identify Basper and say, these are the ways it is distinct. And this is how, if you're growing something you don't know is it Casper or Basper, you can use this key to figure that out. That would be a great skill for us to have as a community. Is just my initial thought about this, that this foraging skill, this general mycological ability and basic practice in mycology, we could transfer over into all these cultigens that we're developing and growing to just give us, right? That's what they're doing in cannabis right now. These guys in the dispensaries can talk ad nauseum about every little cultivar and this is this and this is that, and it's got this much THC and this much terpene, and I'm going to give you a 5-hour lecture about this terpene and that terpene. So obviously they're going very deep on this, and I feel like that is maybe a next step that we could all be taking, is getting a little bit better at describing all the cultigens, all the varieties, all the isolations, all the new crosses, particularly, you're developing these new crosses, and you want to be able to describe them, especially as you go from filial generation to generation, and be able to, once the fruit is gone, then also look back at how they've changed. Are they changing? What's staying the same? Right?
Yeah.
Is that making sense?
Right. There's two major problems straight off the bat. Number one, environmental conditions.
Okay.
As we were talking about yesterday, my patch of bluets, bluets, which used to be Lepista nuda or Clitosabe nuda. Okay. So again, there you got a name problem already too. Lepista and Clitosabe they're basically the same genus, or they used to be the same genus, depending on what book you looked in. So even professional mycologists, one book you would see Lepista, which is generally like a sort of more pinker spore print, and then you have Clitosabe, which was traditionally a white spored mushroom genus. Okay. The problem is some people didn't think that was significant enough. So here's our problem. We as a group have to come together and decide what are the significant characteristics. So just like DNA has, here's why, believe it or not, ACGs and Ts are what we call discrete. They're not a continuum, right? It's either an A, it's a T, it's a G, or it's a C.
Or a C.
That's it, right? An A is not going to ever be a T. Now, cream to me might be beige to you.
Right.
So here's where we got a problem, right? Because every cultivator is going to have a different set of criteria. And to be honest, maybe their eyes are different. If you're red-green colorblind, you know, you're going to have a problem sorting out russulas, which there's a lot of red and green russulas, or russulas, as Alan says it. That's going to be a problem. And if people don't use the same criteria, you know, maybe someone thinks that the gill attachment, for instance, is very significant, and maybe one time you have a grow, it's a little bit too dry, and that cap expands, and maybe the gills separate from the stem, you know, the stipe.
Right.
That's going to look different in a different environmental conditions. Color is dependent on the, you know, the light spectrum. We know not so much, but, you know, obviously, like mushrooms that are exposed to more light are going to get darker caps.
Right.
So if I'm running, you know, 5,000 K LEDs and somebody else is, God knows why, running metal halides or something, you know, high pressure sodium above their grows, you know, next to their weed, maybe, you know, they're going to have different colored caps. And then you get into things like the albinos. Like, how are you going to describe the cap color on you know, I don't know, even the leucistics, or leucistics, however people say it. How are you going to describe that? How are you going to describe an albino A+, you know, like, I don't know. Like, I've grown them about 6 times, and every time they've got like slightly different caps, even from the same clone, you know. It's like, and then you start talking about microscopic characteristics, you know, and then do people have the right measuring device, the right micrometer? Are they measuring a spore the correct angle and, you know, vantage point?
Right.
So the list goes on. And to be honest, we shouldn't be so hard on ourselves. Professional mycologists have been arguing about this for hundreds of years. I mean, these guys, Fries and Persoon and Alexander Smith and Rolf Singer and all these people. Rolf Singer he's like an icon. He is probably one of the most controversial mycologists of the 20th century, because he basically said F everybody and like rearranged. He's got one book. If you want to get a book that really will make your head spin, it's called The Agar in Modern Taxonomy. I think he got up to the 5th edition. It is a monster of a book, and it will tell you the nitty gritty details on, like, when you see at the end of a name, you'll see like psilocybe, and then you'll see maybe in parentheses, you'll see like Fries, FR. It looks like fries, but the way you is Fries. And then outside of those parentheses, it'll say Singer. Singer is a reference to Rolf Singer. So Rolf Singer was the one who basically revived the word, the genus psilocybe to include our cubes. Unfortunately, we've got other problems with taxonomy and the actual, what we could get way into this, but the type species of psilocybe is Semilanceata, or the Liberty caps, which are a tiny little thing. So the type species of the genus should be representative of that genus, and it's not, which is a problem.
Oh, so you have not only like the voucher specimen, the holotype. I've heard about 6 different words used to describe the same thing, which is the first one we use to base everything else on. You don't just have that for a species, you also have that, you're saying, for a genus, or maybe even further back, or no?
I believe there's types for the families too, but at this point, the type for the family would be a bit ridiculous.
Oh, so it's mostly genus and species that you'd have a type for.
Yes.
They're sort of like the model. It's the goody two-shoes species that most embodies everything about that genus, is the idea.
Exactly.
And you're saying Liberty caps is that for psilocybe, but it's sort of like a shitty example for psilocybe.
Yes, exactly.
Okay. But that's Singer's fault, you're saying?
it's history's fault.
Okay.
Because the genus Psilocybe was typified with that as, so when you make a new genus, say 100 years ago, you would have designated a type species, and the person that designated that genus, you got to go back and look at the nomenclator, as they call it. It's a list of like who named what date, etc.
Okay.
And it gets messy real quick. It's not the kind of thing you probably want to do unless you're unless you're publishing papers.
Unless you're trying to get them to be published, yeah.
So these things are all like, they're all quite, and then you get to opinions. There's opinions. People have different opinions. Maybe some people think the color of the cap is not important. Maybe somebody who, like me, went back to the same patch of bluets and picked them year after year after year at the same location on the University of Michigan campus under that one pine tree or that one group of mixed hardwoods. They look different in June, July, August, September. They would come up month after month, and they always looked a little bit different. But I knew they were the same mushroom, it was the same pile of wood chips, the same trees, the same place. So I don't know. We shouldn't be too hard on ourselves. I don't know. And I know a lot of people like to jump to like, well, DNA will solve all of these problems. But as you know, DNA gets deep real quick. I just saying simply ITS regions or sequences are going to solve all these problems. If it adds another layer. So it's kind of like when you've got,
like it solves one problem, but maybe creates 3 other problems.
Yeah.
Or at least complexifies the situation.
Yeah. Well, when you've ever like stripped paint, if you live in an old house and you go start stripping paint off the banisters and you see there's like 6 layers of different colors under that paint.
Right.
Like it's like maybe you've got a DNA sequence, but you inevitably have to look back into the history, the names, and what other people did, because it will have no context. You can't just like lay out a DNA sequence and be like, this is my new species.
Right.
You have to look at all the other things that are surrounding that species.
Right.
This
brings up a thing we talked about that I had wanted to talk about on our first podcast, so let's talk about it here. Ed introduced me, maybe at some point in my liberal arts education, I had been presented this idea, and I think that some of the core of it was familiar to me, but I didn't recall this term, and he was talking to me about it as it relates to species differentiation, biological species model. Do you want to talk a little bit about what that is, why it matters, how it came to be, just a general overview of that and how we might use that in what we're doing?
Yes, that's so that was one of the main points of my PhD, was basically to sort out how the overlay of the biological species. So I had 3 components, which is the morphology, the DNA sequences, and the biological species concept. There's other species concepts. There's, I believe, about 17 if you look on some of the websites.
Oh, that's it? Only 17?
Yeah, like
okay.
I believe there's a Stanford website that like lists 17. So you've got things like a geographical concept. So for instance, like if something grows in Europe, a lot of the European names were applied to North American mushrooms, and we know very clearly now that they're not the same. Like the boletes in North America, and the boletes even west of the Rockies and east of the Rockies, the boletes, like the porcini, the king boletes, they're not the same. Chanterelles are not the same. And like I've been to Europe a few times collecting mushrooms for whatever, and they're not the same. You see a mushroom and you think, oh, wait. And then you pick it up and you're like, no, that's not right. Something's wrong. It's not even subtle things like the density of the mushroom. You'll be like, this doesn't feel right. And you get, I hate to say it, but you get sort of a vibe, even with our babies, our cubes. I don't want to sound all hippy dippy and stuff, but you can get like a certain vibe from some of them, like, like if they're happy or not.
Right.
Like, I don't know. Maybe I've eaten too many babies. But
I was gonna say, how many did you have at that point?
They
Yes.
talked to me. I had to
Yes.
try my new crosses.
Yeah.
No, that would be a bad idea because I got like 6 of them like ready to go.
Yeah, not all at
But
once. Not all at once.
yeah, the ecological species concepts. If you want to talk about like I don't know if it was the different times of the year, certain things will fruit very early in the spring or maybe midsummer or fall. And that sounds like maybe it's irrelevant, but it's not, because remember, that's when they're breeding. So if you have a mushroom that fruits, it's like frogs and toads and salamanders. If a salamander's in a different pond or a different stream or whatever, if they're surrounded by forest and separated by a mountain that they can't crawl over, they're gonna get so the basic idea of a biological species, so we'll just get to that, is reproductive isolation. So when you become species, it by default means that you have become reproductively isolated. Okay, so even if you go to humans have not been reproductively isolated from each other. Even if you, and neither have dogs or cats or horses or cubes. They just simply haven't. So evolution in a natural context takes not millions maybe, but tens of thousands of years. And to become fully reproductively isolated. Remember, reproduction is the primary goal of any living organism.
Right.
So for that to change and for you to become reproductively isolated, i.e. a new species, that is a very, very, very significant event. Right. If you went to another country and for some reason couldn't, reproduce with the females there,
that would be terrible.
Kind of a bummer. Yeah. You show up on some awesome like Pacific island and there's some, humans there and you can't, mate with them. That would not be very fun.
It would be weird. Yes.
Yeah, that would be weird. But even that island, again, these people that are isolated on islands in the middle of the Pacific or maybe Polynesia or wherever, like for thousands of years, they are still reproductively compatible with human, like the rest of the humans. We're all the same. So that emphasizes the fact that the morphological or the phenotypic differences in humans is irrelevant. It's obvious. We can all procreate, and we
like you can have some like indigenous Mayan that lives in Mexico and she's 4'2". She's just, absolutely this ancient human Mayan person. And you're like, wow, but she could date David Beckham and they could have kids.
Yeah, no problem. How about Dennis Rodman? She could date Dennis Rodman.
She could. And that actually wouldn't be surprising for him.
Hopefully the baby wouldn't come out all tatted up with
If you literally told me, did you hear Dennis Rodman is dating a Mayan chick from Tijuana? I'd be like,
I wouldn't doubt it.
Oh, cool. No. Yeah.
He's probably dating North Koreans. I don't know.
But that metaphor that you've used the humans with me, and that one's powerful. Or you've used dogs, where it's all caninus. What I don't know what the genus and species for dogs is. I can't remember either. Caninus domestica or something like that, probably. Something like that. I mean, I have a little wiener dog, right? A dachshund looks radically different than an Irish wolfhound, and yet with maybe some physical assistance, right, a male dachshund could inseminate a female Irish wolfhound and theoretically give birth to some sort of weird babies. I mean, that's how we have labradoodles right now, right? Like we have all these new boutique very expensive breeds of dogs that are just two other varieties bred together to create the new variety. And that's really what we're doing, right? As far in the cube community, that's really what we're doing. In the gourmet community, it might be like taking one variety of dog, like taking one dachshund and just how can I make it more and more perfect? So the gourmet guys are kind of more like Westminster Dog Show. I want the perfect of whatever I'm growing. Whereas a lot of the cube guys are like, I want to create a monster, right? Like they want the weirdest, most different, unique thing. But it's the same concept where you have all this morphological variation, different phenotypes. But at the end of the day, what you're saying is you put the spores together and they can germinate. Biological species concept says that then that's the same species, is what you're saying.
So but we have to remember what we're doing is also very artificial, right? So when we select, so what we do is called artificial selection, right? In nature, maybe an Atalensis would never mix with a Mexican cube. Mm-hmm. They're geographically isolated. Right. So we got to be a little careful because what we're doing is we're taking things that could in nature, if given enough time, they could speciate. They could become different species, but it goes back to the same thing we were talking about humans and dogs. There's basic genetic necessity to mate. So organisms, it takes a lot to get them to be reproductively isolated. And
a lot of time and a lot of factors have to fall into place. A
lot of factors, yes. Like it could be hundreds of generations. I was laughing, Mike Denver. You're not becoming reproductively isolated. You have to reproduce first before you become.
Wait, man, I must be not getting any of these.
It's in the
my live stream must be gone. Holy crap! I wondered for a minute. I was like, "Are people not commenting anymore? What's going on?"
I'm in the middle one that says comments, and I can see a bunch of people commenting.
My last comment was tip of the cap saying Caesar mushrooms in Europe versus Amanita jacksoni. So it's probably I don't know what just happened, but mine's gone.
Pausing or something. Yeah, mine's doing weird stuff. Who knows? So somebody mentioned inbreeding too. Inbreeding is another thing that we do, and this is what a lot of the land races. Again, we're getting into tricky terms here that sometimes get used in the plant world that don't necessarily apply to fungi. But in the plant world, a lot of the breeding that they do is with inbred lines. There's some called homozygous or pure breeding lines.
Right.
In the tomato world, you might call these heirloom varieties, and in maybe the cannabis world, they would call them land races. The problem with our cubes again is we don't really know what a land race is because we have domesticated cattle, and cattle spread the spores of our cubes everywhere they go. So that Mexican cube might not be Mexican; it might be African. Wherever those cattle were, or whatever grass they were eating, or whatever boat they were on, or whatever, we can't really tell. There's another concept called molecular clocks that we could get into later, if we get into deeper levels of taxonomy. When you go back in time, there's ways you can use DNA to basically calibrate the evolutionary process. Again, there's a lot of assumptions, and we probably wouldn't do it within a genus, but when you start getting to family and order levels, you can, those, I don't know, like those old Cosmos with Carl Sagan and the Neil Tyson deGrasse. Well, they'll put a 24-hour clock, and they'll say, well, the first second, we're basically in the last 5 seconds of that 24-hour clock, of the 4.6 billion years of Earth's existence, or whatever it is now. So that's a little tricky too, but that's just another concept that people might be interested in later.
Because I think about that. I think the first person to bring it up was Kyler Diamond brought up. I can't remember what it was about, but it was about the idea that there had been papers that had suggested that the species in one region was most likely genetically connected to a species from a whole nother continent, and then there was a hypothesis about how it got there and all that, and archaeologists and all that. I'm sure as DNA plays more of an issue, and as there's a paradigm shift in how academic archaeologists start reframing stuff once they've been presented more DNA information, I imagine we're going to start learning where things were and where they came from, just simply even using the DNA.
Cool. Well, Okay, this is kind of related to what we're talking about. We're overlaying these concepts. So you have the paleontologists that they can look back at the bones of animals, and for sure you can see that South America and Africa were connected to each other, right?
Right.
So back in whatever you want to call it, Pangea, Gondwana land, or they got different names for all this stuff. And you can see in the plant species and the animal records that there's, things like marsupials, like how marsupials are distributed throughout the world. If you go back, those things correlate to plate tectonics, right? So one of the things I studied is called biogeography. What you're referring to is biogeography, which is, as the name implies, the geography and the movement of biological organisms and how they've distributed themselves through, like millions, hundreds of millions of years of. So in that case, obviously something that lives in Australia is going to speciate and be reproductively isolated because it doesn't have a choice, right? Now you've got places like North and South America, then you've got like the Bering, up where Alaska connects over to Russia or whatever it is. You've had those historical events, and so you can draw all kinds of correlations. Not only that, animals and plants and people. Right. One of the kind of like funny things that my girlfriend always asks me, my wife, girlfriend, whatever. She says, why do Native Americans, they look like Asian people? And I'm like, because they are. Right. Like Native Americans, like that guy down that Aztec, that dude's Asian. Right. Believe it or not. That little 4'2" Mayan woman, she's Asian. Like she's like closer to Chinese people than probably Chinese people are. Wow. Because they diverged again. So you get what they call bottlenecks. A bottleneck is basically where you have some event. It could be a boatload of people. It could have been a Viking boat landed somewhere, I don't know, in England or whatever, and raped and pillaged or whatever they did and decided to stay.
That's what they do, Ed. That's what they do. If you're not raping and pillaging, then how can you even call yourself a Viking?
Exactly. All those Norwegian black metal rapists. Anyway, sorry if there's any Norwegians out there. But that's when species, that sometimes they call it the founder effect. Say, for instance, if you had, like in the Galapagos Islands, if you have, maybe only a few individuals or a handful of individuals that make it to a new habitat, and then they get like isolated by, again, maybe an ocean or, weather patterns or whatever, they will also, again, start to speciate and become all the little, Darwin's finches and all that stuff. So you've got little islands that have all these ecological niches, which again is another pressure to speciate. So what we do as cultivators and plant breeders and all these, or dog breeders or whatever, we select for what we like. So we got to remember that too, is that we're selecting for things that we want. So in our head, what is a good characteristic and a bad characteristic is pretty subjective, right? So we want stronger cubes, we want larger yields, faster flushes. That doesn't really mean it's better. That's just what we want, right? Like it's.
Or some of us don't care about that, and we just want another one. Show me something else. Oh, I have not seen that. Let's grow it.
I like cuteness. I think cuteness is what I'm going for now.
Okay, dude, you're gonna love some of the stuff I sent you. Then I sent you a couple. They pack a punch, but they're cute. Yep.
I know. Yeah, I've grown some cute little white ones that are. It's funny because I hear comments now. I didn't know the phenotype when I first grew them, and I'm like, what the hell is wrong with these guys? On your hat there. That was like, why are they so small? Like, did I do something wrong? No, it's like now they're supposed to be like that.
Yeah, man, it's crazy. If you grow like some DC Max or some just some old school Melmax, they're all this big. They look like prehistoric mushrooms, and then your next tub is like some ghost. You're like,
it's weird. I'm like, did I put too much like something in my substrate? Yeah, dude, I got an albino Cambodian now that's pushing like a foot tall. and it's like crawling out of my shoebox, like pushing the Martha tent, like the thing, the door, whatever it's like pushing on the door. I'm like, oh, so yeah, Alan, you're preventing me from harvesting, man. That's you're I'm abusing my babies because you didn't show up, buddy. I'm supposed to be listening and harvesting, not talking.
We're cutting into Ed's harvest time here.
It's all right. They'll just spore you later. Well, not those guys, but I got all those gnat crosses, man. They're gonna look great.
So let's talk about those. Since I've seen the pictures trickle onto Dave's group, and you sent me a couple pictures. I'm assuming you're using your ghetto cereal dilution, or are you using a more traditional cereal dilution method?
I wanted to talk to you about this, and I know opinions varied, but the one video I made where I shake the heck out of the little glass, like 100, that works really well.
Okay.
So what I do is when I put them on the plates, the spore dilutions, I just use like a tiny little drop with the pipette, and I make about 20 little, like doot, 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 doot. And I'd say maybe about 4 or 5 out of 20 will grow something, and I have gotten really, really good success rate getting monokaryons from that. Up to the point where I'll isolate like 10 little putative monokaryons, and like the first 5 will be all monos, and I just like don't do anything with the other 5. Like I just let them grow out and form a dikaryon, just for backup. And so yeah, that method, just putting, I really I might redo the video, honestly, because to make it a little more concise, I think Dave mentioned this, and maybe even somebody else maybe mentioned it, I can't remember, so many podcasts and stuff, taking like a really sharp scalpel and just ripping a little piece of the swab off, like a tiny little piece, and putting it in sterile water and just shaking the crap out of it. Maybe a drop of Dawn, not a drop, like a teeny little bit, like a pinhead Dawn dish soap or the Tween. Shaking the heck out of it and letting it sit for maybe a couple minutes while you sort things out, do your mirepoix, and then shake the heck out of it again and just get that dropper, and just use 3 or 4 plates and drop like 20 drops on each plate, and guaranteed one of those you'll get 3 monokaryons at least. And you only need 1, right? You only need 1 monokaryon.
That's interesting, because that's similar to I think it was Julian Matucci was talking to me about that he uses, in lab practices, they have those little plastic loops that when you dip it in a solution, it pulls out like 1, microliter of something, or they have the different loop sizes, but it pulls out a pretty consistent aliquot of substance. He was like, just use one of those, and you're just doing 10 to 20 drops per plate, and same thing. So that obviously works. And then when I watched the ghetto cereal dilution method, with just scraping a couple spores, scrape just a little bit of spore off of a print into a jar, and then shake the crap out of it. Not even 2 days later, I think pretty recently, or soon after I saw that video from you, Dave Wombat had a video where he was like, I just dilute it and I squirt it onto a slide, and I look under the microscope, and if it looks like there's too many, then I dilute it a little bit more. And I was like, yeah, cool. I mean, I'm not publishing a paper. I don't have to have this extreme way of isolating down to a certain amount, and really all I'm trying to do, like you said, is do a bunch of dollops on a plate and hoping a couple of them are monos.
Exactly. And what Dave did, I think I know what you're referring to, he kind of diluted it in the syringe. That's another brilliant idea. To just like, you kind of do this cereal dilution in the syringe. You're not dealing with a lot of vials and crap like that laying around. To be honest, I know I'm going to piss some people off here, but the cereal dilution method, the 1x, 10x, 100x, that's for CFUs, colony forming units. That's for quantifying bacteria. We are not quantifying spores. So when you do a 10x plate and a 100x plate and a 1000x plate, the reason why you're doing that is so you can work backwards to figure out what they call CFUs, colony forming units. So if I give you a sample of, say, ketchup, and you can't take a blob of ketchup and put it onto a plate and count bacteria, there would be way too many. So the idea is you take the ketchup and you dilute it to 10x and then 100x.
Oh, and then by working backwards, you extrapolate the density of. I think I learned about this in microbiology, but I forgot.
So on a 10x, so on a, just say 100x plate, if you see 3 colonies form, then you can I don't know, interpolate, extrapolate, whatever, go backwards. And you can say, well, then on a 1x plate, I would have 300, right? On a 100x plate, I got 3. And then you go back and you say, well, at 1x, so in that original bottle of ketchup, you have 300 colony forming units per milliliter or whatever.
Gotcha.
And that is useful for bacteria. It's not really very useful for us. And to be honest, it's kind of irrelevant, because we want monokaryons. If you want to count spores, you get
thing, sure.
Right. If you want to count spores, you get a hemocytometer. There's little slides that have little grids on them, and that's what you use to count blood cells, the blood, like a hemocytometer, cell right? Meter. So it literally means blood cell counter. Yes, that's what it means. Yeah. And we don't do that. We don't need to do that.
I mean, is there really any instance, because from what I've read, there's never a descriptor of specifically how many spores a fruit ejects or a species ejects. I mean, it might say, heavy or light or something like that. But so is there ever an instance where you specifically need to count spores?
I can't think of anything in home mycology. There's estimates, like they say puffballs will make billions and trillions of spores.
Just to kind of know.
But for home mycology, unless you were going to prepare a commercial product that you were going to sell as an inoculant, I don't see why you would ever want to count spores at home.
Gotcha.
So yeah, I think, I'm sorry, I'm seeing Dave's comments here. I respect Dave obviously quite a bit. And like I said the other day, sorry Dave, I'm kind of stealing or reproducing. I'm just like, what is that? Imitation is the most sincere form of flattery. Is that how it goes?
See, I can't see any of these comments. I don't know what he said.
Oh, there's a whole peanut gallery going on over here. I don't know why I can see it. I'm not really sure why.
No, I usually can. It must just be some glitch with. I wonder. I'm trying a new thing. StreamYard, the platform that I use, it's trying to be more like Riverside FM. That separately records. So right now it used to be it's only recording this whole broadcast together as one audio video file. And they just added the ability to record your video and audio separate from mine so that you could play around with it later. So I want to give that a try tonight. So maybe it's got to do with that. I don't know. It's just a glitch. I'm sure it won't be like this all the time. But
yeah. Mine looks a little bit different than it did last time too.
Okay.
But before we move on, I just want to tell you guys out there, since I was on the podcast and since I've been posting some of these videos, you guys, this is and again, I'm sorry if I'm messing with anybody's bank or cash flow, but making monokaryons is not that hard. It's really not that hard. Yeah. It's pretty straightforward. Watch every YouTube video you can find, and even watch the ones about microbiology where they're doing proper serial dilutions, and they'll do the math and all that stuff. You don't need to do the math. The basic idea is you want to get a single spore in a single drop of water. The other thing about actually doing serial dilutions is maybe they're going to be rehydrated a little bit. If you got an old spore print and an old swab, you put them in the water and let them sit in there for 15 or 20 minutes before you put them onto Agar.
Okay.
It's really not that tricky. It's just numbers. Right. So you just basically have to burn, like if you got one print, imagine you're going to use at least, let's just say 5 plates. I guarantee if you just do, even if you mess up the dilution, even if you use the 10x instead of your 100x, you're still going to get monokaryons. And in a completely different discussion, a lot of times when these spores germinate, they grow out concentric radially. Yeah. So if you have a spore here and a spore here, they're going to grow out like this. So what you do is you get the tips of those hyphae. So when you're trying to isolate a monokaryon from even you get a little spot on a plate, it might be almost, maybe a quarter inch or a half an inch big. Try to isolate from the edge of that colony and you'll be surprised. Right. You might just get a monokaryon.
I see. I think I read something somewhere where they were basically saying the very leading edge of a growth colony frequently will still be haploid. Yeah, it won't be.
Yes, exactly. Both, and some of the techniques that are currently being developed by some of the probably some of the people in the chat rely on this. That the leading the hyphal edge is where the monokaryotic mycelium is going to be, whether it's a monokaryon or it's a mixed culture.
Now, man, I got to tell you, I got a problem because I just so there's this guy on Facebook that I like a lot. He seems like a real earnest guy. He's also a beekeeper. For a while, he had two different names, but I believe his name is Broccoli. And he had just a post not too long ago about trenching on a plate. That if you create trench
sequestration,
that the hyphal tips grow across it, and it gives the mycelium a chance to get ahead of the bacteria. That then the bacteria have to kind of dip down, and I have to imagine they could still make it across. But anyway, the idea is
I just did it. I just cleaned up a rusty white culture. It works brilliantly. I made the gap.
I've done it before. It does work, but I have to imagine that the timing is still important, right? Yes. Like if you wait too long, eventually I believe that bacteria could catch up, and then so you have to get your timing right. But anyway, he posted that, and I was doing that whole thing, and I was just like, yeah, man, these the timing of a lot of this stuff is more important than I think anybody talks about.
Yeah, exactly. When you
so when I was cutting that, though, here's my question. Sorry, I got sidetracked in my ADHD. When I go to cut it with my brand new, fresh out of the package, number 11, it still wants to it doesn't necessarily want to cut, and it pulls some. Do you know what I mean? It's not like a super clean surgical cut.
I know.
And I'm just like, so I want that very leading edge, but sometimes I feel like I'm getting more than I want. I don't know what the secret to that is that I got to start farting around.
I might have a tip for you. So cut parallel to the hyphae. So when we've got the radial hyphae here, cut this way, and then cut where the hyphae isn't, and then go back before the hyphae and just jab straight down. And then I'll jab down. So I'll do like two cut. Let's see if I can do this. So cut, and then cut outside where there's no growth, and then go back here and just jab straight down and pry it out. So use the very tip of the 11 and pry it out. There's actually two reasons for that, because you also want to, if you're fiddling with, I mean, sometimes I cut off it must be maybe a 1/16 of an inch big piece. And also sometimes I'll kind of get in and almost like you're scooping ice cream, get under there and kind of try to pop it up. And sometimes it works well, sometimes it doesn't. It really, I think, depends on the auger, how whether it's 1.5 or 2%, or sometimes I go a little higher, how stiff the agar is and the ambient room temperature and all that kind of stuff. But yeah, I usually try to get the three sides that are kind of clean cuts, and then the fourth side to be the messy one. And I usually try to pop it out away from the growth, because here's the problem. If you're doing a monokaryon and you pop it and it lands back on the center of the colony, you could contaminate it, and what you think or what you just had popped out was a monokaryon. Now it's a dikaryon, because it just popped onto another, and it picked up one little tiny piece of hypha from another colony. And so that's one thing.
Yeah, that's what. So Julian Matucci just posted his DDK protocol that he used based on this other published article, and he was saying in that, and then in talking to him, he was like, so you think you got a mono, you have to let it grow out for like 3 to 4 weeks, because some of them might be doing their best to look like monos, but they might ultimately end up being dicaryons, and you just got to give it that time. And I had one of those where I used your little Vaseline trick, and I got 3 monos, 1 converted though. So now I only have 2 monos, but I'm like, okay, so lesson learned, a mono will never stop being a mono if it's a true monokaryotic growth. So sometimes it's worth letting that second plate really grow out and double check, because man, that third plate,
unless you have homophilic species or multinucleate species.
sure, but I don't.
I read a paper yesterday about Caprinus, and it's it's a mess. It's like,
and it does
Caprinus cinereus, which is a model organism that it does weird stuff. But we don't need to talk about that. Let's stick with our cubes.
That's not. Man.
So
I'm not growing Caprinus.
Yeah. Don't want to.
So that's a good technique, and I vaguely recall seeing that when I first got into mycology. Man, I read a lot of shroomery, like probably too much. It probably just confused me and slowed me down. But I think I remember seeing that. So then when Brock Lee posted that, I was like, oh yeah, man, I forgot about that. And every once in a while, I get wet bubble, or I just get the where you can tell the leading edge of the mycelium is just a bit behind some very lighter, almost film-like, like something bacteria. Like, oh, dude, I got it. I usually just chuck those plates, but I was like, man, I got to just do this trenching and see if I can recover some of them. So that's working out.
Another thing, sometimes people will take a agar square and flip it over and then let the mycelium grow up. I've seen older things where they'll take like another scalpel and they'll make like a little envelope and put their new transfer under and let the mycelium, because the mycelium will grow up, and then you can isolate from the top surface. That's why, there's some tricks to it. I mean, worst comes to worst, you can put antibiotics like gentamicin in it, but I don't like to do that. If you've got, I don't know, I luckily don't have that problem anymore, but I know a lot of beginners really, really struggle with contaminants. And
you sent me a cool article. I assumed we would do a podcast eventually just on it, but if you want to talk a little bit about your just basic 101, besides obviously cleaning and sterilizing everything all the time. I don't know if you have some general contamination thoughts.
Yeah, the one thing that I've learned over the last few years, pressure cook longer. There's 3 things, three old wives' tales, or whatever you want to call them, that I probably learned from the shroomery too, was don't overcook LC. It'll caramelize and the fungi won't grow. Bullshit, right? Fungi have enzymes that degrade polysaccharides. Cellulose is a polysaccharide. Like starch is a polysaccharide. So I was a little confused when I was seeing this, like, well, sugars will caramelize and the LC won't grow. And as you know, if you've messed with LC, there's like 100 recipes. Everybody's got their own maple syrup, honey, sorghum syrup, blah, 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 blah. You know what my LC
now Jemima?
Is? No, some bang. Yeah, a couple sausage links, some Jimmy Dean sausage.
Just 2 sausage links. That's for just thick.
I don't
know. What do you use? I want to hear what your recipe is.
0.5% LME. That's all. It works great. Wow. But I like kind of the more wispy stuff because I use 18. I don't use LC all the time. I actually kind of vacillate back between LC and straight agar, but I go back and forth depending on my mood and the culture. If I've got LC that's ready to go, I'll just use that. If I've got a fully colonized agar plate, I'll use that. But just straight up LME. Forget all the other stuff. Peptone, useless. I'm sorry, people out there selling Peptone. I know some.
Man, don't hurt my feelings. I just got some LC from Philly Golden Teacher, And so my recipe is corn syrup and a little bit of LME, which I got from Fungi Guru. And yeah, I've heard other people say that they don't. So the old thing I heard was like 2 to 4% sugar, right? And I've heard other people
way too high.
I've heard other people like you say 0.5%. That's by weight or volume, that's more than enough.
Anything above 2%, I think, is ridiculous. It's a waste. It makes the LC too dark, and it also, I think it like sort of. Somebody said before, it gives the fungi like sort of diabetes or something. Like so one theory I heard, which is probably true, is that the less sugar you give it makes it active. You know, like kind of when you see rhizomorphic on plates, usually the lower nutrient plates get rhizomorphic growth more readily. Same kind of idea. You want you want the fungus to be metabolizing those sugars and stuff. But again, when you go from like a pure refined or semi-refined sugar, like a fructose, glucose, honey, even LME, right, is a lot of sugar, and you go to whole grains, the fungus needs a bit of time to acclimate. So if there's anybody developing new liquid inoculum or LC recipes, not inoculum, LC, liquid culture, then I would like kind of hesitate to go to the pure sugars, maybe throw in a little bit of grain water or something like that. If you really want to like using like, just save like a half a liter of your grain boil water. And I was using popcorn, the water, because popcorn's pretty clean. So I was just saving the water from boiling my popcorn and just putting like this clear, really super clear, like Korean corn syrup in it. But I was only using like, I think 1% maybe, and it worked great. But I don't know. Some people like thick LC. I do not understand this. Why do you need thick LC?
Because we're Americans. We're Americans. We want it big. We want it thick.
Dude, man, this is the thing. Like the dark black spore syringes, the MSS. Like, why do you need a black dark spore or swabs that don't have enough spores?
Right.
I don't understand this. I hate to say it, but it's people that maybe don't understand that if you can see spores on a swab, that's like 10,000 spores. And
you have too many at that point. Yeah.
Right? So the LC. I mean, I used some of my my LC last week, and the bag was colonized. I only used like 1 cc and a bag of popcorn. 5-6 days later, it was fully colonized. I usually shake it with LC. I shake it after the second day.
So now, are you still doing? Are you mostly just doing the straight LME, at 0.5%, or are you also sometimes doing the grain water just as you have it?
Now I just do straight LME because it's easy. Okay. It's powdered. My freezer was full of like popcorn boil water, and I just got sick of shifting it around in the freezer. Yeah. One of them fell down, like a 1.5 liter frozen bottle fell on my foot one day, and I was like, I think I'm gonna get rid of that.
Yeah.
So powdered LME, dead easy, man. Just mix it, give it a shake, pour it into your bottles or jars or whatever the heck you're using, and just whack it in the PC. I do an hour and 15 minutes. My PC only gets to 11 psi, like 70 kilopascals. So
I'll have to do the math on that, but
it's basically longer is better. So going back to the caramelizing stuff, I read stuff on the Shroomery over and over and over again about caramelizing grains. Do you know how long you have to cook whole grains that are properly hydrated to caramelize them? It's like 6 hours, 8 hours. You'd have to go to sleep and wake up the next day, and your PC would run out of water.
You'd run out of water before that anyway.
Right?
Yeah.
So it would be irrelevant. The whole thing would be, your bags would be melted, and your house might be on fire. Okay. So I don't know. I hate to say it. I love all the stuff that's out there, but a lot of it, and I hope with the help of you and other people, we can maybe dispel some of these myths.
Sure.
There's a lot of misinformation out of there, and it's a lot of old wives' tales. And I hope to think that like people aren't purposely spreading misinformation, but I have a feeling they are.
I hope not. Here's what I think, because this comes up all the time. So the more recent thing that's come up is the I have a small segment of friends who basically say, bro, why are you fucking with that Dr. Mike's MPG Plus? I mean, you were growing mushrooms before. What the fuck do you need it for? Like, what's wrong? Are you just trying to make your life more complicated? And that's the problem, right? Like, I have one guy telling me when you cook your popcorn, you can have no more than just an occasional cracked kernel. And then the next day somebody posts, oh, I boil the fuck out of my popcorn. They're all split down the middle and my mic rips through that shit. The problem is fungi, it doesn't get ideal situations out in nature. And all it wants to do is eat and break shit down and keep going. So it seems like the mycelium can kind of figure it out, whether you're doing, quote, the right thing or the thing that doesn't even matter in the first place. So when I get somebody who texts me and says, I heard you talking about azomite, should I do it? Does it really work? I'm like, I don't know. I'm growing mushrooms pretty much regardless of whatever I put in anything. But I love playing around. I think the more I do it and the longer I do it, I'll get the sense, because I can't ever do a cohort sample size of 200 tubs to decide if something really works or not. So I'm just going off I'm going to trust my intuition over time, because I'm fairly good at observing what's going on with my grows. I think that's why things keep going, is a guy says, man, it's got to be like this, and this is what works because my grandpappy taught it to me, and I grow big mushrooms, man. And it's like it's that old science thing of correlation is not causation, right? Like, you can correlate two things together. It doesn't mean they cause the other thing. So it's easy to go, I don't know, man, I've been growing. And I could be doing this with MPG Plus. I don't even know. But my sense is that it is doing many of the things that it's advertised to do. But I think in 10 years, as I keep going back and forth, I will continue to have a better sense of that. And as long as you keep, this is my whole deal. If all you do is jump into Mycology and you find some proven texts and you do that for the rest of your life, you didn't learn how to do anything other than grow those mushrooms. And for a lot of us, that's not the whole deal. Yes, we love the experimentation. We love the trying new things. I mean, dude, in my Discord server, as you know, like, people swear by the fuck around and find out ideology. Like, that's that old school primitive caveman scientists, right? We just want to fuck around. Many fuck around, not all find out, and we like to find out.
There's every time somebody says that, I think of this old Wesley Willis. There was a rapper who was had some mental issues, who would rap.
Man, I haven't heard that name in forever. Now I know you're from Michigan, because in Michigan, that was a thing.
He had some pretty interesting songs, like but cheetahs having, like, a lot of bestiality based songs about honey badgers and male hyenas and things.
He
like
made
that.
Cool Keith look real mainstream, that's for sure. Yeah.
Exactly. Yeah, Cool Keith was like, Cool Keith was like Michael Jackson.
Basically, Wesley Willis. Dude, you got me. After this podcast, I'm gonna have to go listen to me some old school.
I know. Yeah.
Wow.
Llamas whooping a llama's ass.
So you okay, we gotta get back on track, or I'm gonna laugh about Wesley Willis for way too long.
I know. All these lyrics are coming into my head now.
0.5% by weight, it is all you're running for your liquid culture. You're sterilizing a long time. Don't be afraid of caramelizing. It's not likely. You're not caramelizing grain for sure. And you're saying that even if I used sorghum syrup or corn syrup or whatever, I'm not likely going to be caramelizing it unless I cook it for a really long period of time. Yeah.
Yeah.
And even if I do, you're saying the fungi doesn't give a crap anyway. It's going to eat it.
Exactly. Because all you're really doing with LC is trying to just grow fungus until you get it onto grain. That's the only thing, right? It doesn't have to be pretty in the LC. It doesn't, like, I see everybody posting these, look at my LC.
Yeah.
It's like, yeah,
I do that,
cool.
man.
I do. No, I do too. But being fair, the first couple times I did LC, I was all about like, look at my magnetic stir bar. And now, you know what I use? Like empty like pasta jars and like marbles that I bought from the like kids toy store, and like shitty like airport lids that are like half rusted. No, I do it a little bit better, but I'm exaggerating. But the first few times it's fascinating.
Yeah.
And you know what I wanted to do? I'm like, you know, I could make like 100 liters of LC and inoculate the entire campus. Like every piece of wood on every building. I'm gonna go get like a super soaker.
That's the way to do it. You wouldn't be the first person to do that. I've heard many a folklore tale of people doing that stuff. Yeah.
Definitely. LCs, these people that are like, I'm gonna get a bunch of spores and throw them on these wood chips. Nah, waste of time. Make LC and then just go dump like a 5-gallon bucket all over everything you see. And it's cheap, right? That's the other thing, to be honest. LC shouldn't be expensive, right? A lot of our hobby, when people start adding things that they don't need, you increase the price. Like your agar plates, man. You know what? It's agar, a sugar, and that's it. Like you need, and water. That's all it is, right?
Yeah.
You don't need all this fancy stuff. And again, I hate if I'm pissing somebody off who's selling this stuff, but you know, I made at Dave's like inspiration. I made like ramen noodle agar. And like I don't know if he saw it, but I posted one. I actually put whole basil leaves.
Oh, I didn't see that. I saw your ramen noodle one, but
yeah, because I joked about making pesto agar, so I put actual whole basil leaves in the agar. Sorry, guys, I say agar. It's Michigan, whatever, agar. We have these things called augers, which you use to drill holes in ice for ice.
That's why I prefer agar, but yeah,
I don't know. But
otherwise I feel like I'm gonna go ice fishing.
Yeah, exactly. But yeah, I put whole basil leaves in there. Pasta water. You know what? If you're making spaghetti, save the water. You know what you do? Put 2% telephone brand agar from the Asian food store. Put it in there. Bam, you got agar plates.
Yeah, man. So Tim of Tip of the Cap and Jesse Noller of Humble Fungus were the first two people to talk to me about how you could be training your mycelium, and you just mentioned it too, where they're like, you know, you could be using sorghum syrup in your agar, and then you could be going to Milo and then spawning to bulk, and now you've just, you know, it's all the same shit, whatever little nutrition profile you've sort of stayed with it the whole time. And theoretically, like you were saying, if you're minimizing the time the mycelium goes, whoa, hold up, where am I now? What's going on? And it just sort of like used to things, then theoretically you should be, like you just said, you 1 cc of liquid culture and 5 days later your bag's done. That's freaking nuts, dude.
Yeah, and you know what? Another thing I've noticed is that if your grains are fully sterilized, like 100% sterilized, and the mycelium doesn't have to fight with bacteria, it's like a lot of people say, I'll still run bacterial bags if I don't have other bags of spawn and something smells a little funny, like a little off. I'll still run it in a shoebox, but like if I've got choices, I wouldn't. But when I've noticed the properly people do not sterilize their grains long enough. Simple as that. And I hate to say it, they see all this stuff about Instapots and all this blah, 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 blah. You got to get a pressure cooker, right, man? We all know that. You got to get a pressure cooker. You're going to have to pay that. Yeah, you're going to have to do it. Stop messing around with the damn Instapots and whatever. Like, it's just stop doing it. If you're going to do this, just like get a goddamn pressure cooker. You know, please. Like I've got fucking 3 laying around. Can I give you one? Like, please.
Just guys, just drive to Ed's house in Thailand, in Bangkok, Thailand. He will give you his pressure cooker.
I got like 2 or 3 10-quart ones that are way past their usefulness.
Yeah.
Maybe I'll cook.
I agree, man. I had somebody a couple weeks ago want to buy my sterilizer, and I always interrogate. I always qualify. I want to find out, where they're at. And they not only didn't have a flow hood, but they also did not have a pressure cooker yet. And I said, man, you got a bunch of stuff to buy and a lot of skills to learn first. You just don't even need this. I appreciate that you want to buy it and support me, but man, there's a lot of stuff you need before you need this.
Yeah.
I agree. Yeah. Everybody I talk to, step one, buy a fucking pressure cooker. I don't have money. Don't do this hobby. If you can't save up $150, this is not the hobby for you, man. You should buy a $5 basketball, go to the park, play basketball. Won't cost you any other money ever again.
Yeah. Or the people who won't drop $10 for a Spore syringe. It's like, really?
Right.
Like, how broke are you?
Yeah.
Like,
I will. Yeah, I periodically will just buy from, and I always try to buy from the original vendor, the original person who created it. Just because, man, I don't know about you, but I do a lot of trades and have received a lot of cultures. And from certain people, they're always great because they really know what they're doing. And then other people, you get something and you're like, oh, maybe at one point in time, this was the thing that it's labeled as, but man, I don't know what the fuck this is now. It's not good, or it's not the thing. And, just support the people that are doing the work. It is not a lot of money. Mossy Creek Mushrooms, he charges like $140 for his isolations of gourmet cultures. And he's not even pricey. If you're in the real gourmet, like commercial level liquid culture, they can be like, I think $300 to $500 for a little 10 cc.
That's what I saw. Yeah,
they're money makers, right? If you have just a killer isolation of oyster mushroom or shiitake or something like that, it's gonna make you money. So $300, that's not even a lot of money. If you really have something special, like if you buy Gary fresh from the farm fungi oyster mushroom, it's good. I don't know if it's $30 or $40, but it's definitely worth that much money because that shit's good. Or if you buy iceberg direct from Miss Mush SoCal, it's good, guys. Like, these people care about this stuff, and their prices are really not outrageous. So yeah, $10 for a Spore syringe is like literally nothing.
It's crazy. How much do you pay for your mobile phone bill every month? $50, right. $60? Come on, these are cultures.
I wish that's what I paid for my.
Yeah, you got like a family.
I got a family, dude.
$300. Yeah. I pay like $15.
It's like $200. Yeah.
Yeah, support these people for heck's sake. It's like, come on, man, really? You're paying them to make more stuff that you want. I kind of like gave up on I'm like, I don't know, I'm okay. But like, that being said, like, if you guys do want, like, I don't know if I'm gonna have some interesting stuff here.
We're doing a giveaway. Don't give all your shit away, Ed. Yeah. I want some of it for my Discord.
I'll hold off.
Ed's gonna give away some stuff, but we're gonna do it on the Discord. Also, for those of you who have made it close to 2 hours in here, I will give you a little taste. This will be formally announced soon, but we're going to do a little 12 days of Christmas, which might end up being like an 18 days of Christmas giveaway on the Discord server. And it looks like we got lots of really cool stuff to be given away. Right now, there's, 5 or 6 giveaways going on. Those will end tomorrow afternoon. So if you're not in the Discord, or if you haven't signed up for those, we're giving away free shit over there, guys. Go get some. And yeah, Ed, we want some of your crosses for that. So don't give it all away before then.
I got some wacky F1 spores that again, like that Dave suggested, the F2s, man. That's where it'll be the monocultures derive from the F1 spores, because like we talked about before, that recombination has already occurred, and that F1 spore print swab, whatever we distribute, man, that's where the gold is. That's where all those stuff's gonna happen. So yeah, I had a Shaki that I crossed with a golden halo. The F1, the initial cross between the two monos, was kind of like a little brown one, kind of looked like a brown Shaki. Right. And then I got an F2 monoculture, popped out albino translucent spores, like completely different. And I just had one isolate. And as somebody, I think, sorry, Dave, I keep referring you, but he said that every mono is not created equal. So even if you get if you get 3 monos from one spore print or one swab, you can go down that route.
Yeah.
But you'll quickly realize that the piles of plates, I had to go with 1 mono for each cultigen because it's getting messy. It's getting
too much
real
work.
messy.
Yeah. But if you can get to F1 and get spores, you can just bank that shit, or you can give it away and let other people do the work.
That's the plan.
I think that's the trick. And that's what Dave's doing really well. I mean, first off, Dave is a fucking machine and is growing a lot of mushrooms and getting a lot of spore prints and a lot of swabs. But really, he knows the trick is to just keep going back to spore and running fruit and going back to spore and running fruit. And Yes, I mean, theoretically, by the time you're F2, I mean, off your F1 spores, I mean, depending upon the specific fruit itself, I mean, I don't know if there's a limit to how many genetic possibilities you could get off of it. I mean, you might ultimately have like phenotypic limits, like you might have 1 million different expressions genetically, but the phenotype might express maybe 5 or 10 different ways or something like that. But still, I mean, those F1 spores. Yeah. If you ever hear these old guys, if you're listening, if you ever hear Dave say, "Oh, I found some old F1 spores," get on that shit. You should try to get him to give you some of those or buy them. Do not think, there you go, Ed. Don't think that, so there's 2 games, right? The one game is Ed did all the work, Dave did all the work, Yoshi did all the work, Kyler did all the work, and now here's this, thing that's been created, and if you want to grow that exact thing, buy it from him. But if you want to have fun and you want to play around a little bit, because don't forget, these all have the same medicine in them, you can get an F1 spore. You can get even an F2 wouldn't be bad. But you can, play roulette and see what you get. And
you're not going to get like a white button mushroom out of any of these. They're all they're all going to be the same thing.
And that's really just you got to be willing to play that game. If you want to have that kind of fun. Yeah.
It is really, really fun. I almost feel sorry for somebody mentioned these factories where they have hundreds of monotubs and they grow 1 clone in all the monotubs. How boring would that be? I mean, I'll like after the podcast, I'll probably mix up like six little shoe boxes, maybe 8, depending on how much sub I got. And that gets tedious, but imagine doing that 8 hours a day in monotubs with, I don't know, like a B+ or a hillbilly or an ape all day long.
Right.
That would be so boring. I don't know why you would want to get into this hobby except for money. If you were.
Yeah. Well, and there are those people.
Mm-hmm. Yeah. There are people that just want to be like, entrepreneurs or whatever.
Right.
But I think most.
And hey, you know what? If you're in this community and you just want to make money by being an entrepreneur and you make a great product, good job. I'm happy.
Yeah.
If I don't care if you make really a great product. If you're making a subpar product that nobody needs, then bye-bye. If you don't make it, I don't really care. I definitely love the people who love what's going on and love growing, and I can usually tell whether you're in it for the money or you actually like doing this. It doesn't take long to figure out. For sure.
You ask like 3 or 4 questions and you can gauge pretty well like what they're in it for.
Yeah, I love it when I get even just the cube guys. When I had Wumbo on, and his interest in mycology is much broader than actives, and a lot of these guys, their bread and butter might be actives, but a lot of them know a lot about a lot of mushrooms because there's just a true love of mycology, and those are my people. I definitely like those guys the most, for sure.
Yeah, and it also gives you, when you study other, not even other parts of mycology, but other sciences and have other interests, that you get inspiration from random places that you would never you'd never think, and especially if you think about mushrooms like we all probably do, like 90% of the time, you're walking through the grocery store looking at pasta jars, like, don't really need Alfredo sauce, but I'd kind of like that jar for.
See, man, I can tell you're in Thailand, dude, because I am not doing that. I'm just buying 12-pack of ball jars, but yeah.
It's gotten better. It's gotten way better here in the last couple, I'd say maybe the last year. They started selling 50 ml PP tubes, this big screw ones that'll stand up by themselves. So they're like those centrifuge tubes that you.
That's what I need, man. I hate having to have the stands for them.
They sell ones now. You might want to look. They just started selling them here a couple months ago. They'll stand up. The base is only about the size of a $50 piece, but they'll stand up by themselves. They're really nice.
Nice. So I got a couple more questions for you before I'd like to circle back a little bit on some science, but quick questions. What sub do you run, and how hard do you pack your sub down, and what ratios do you tend to run?
That's really good questions, and I've been contemplating that myself. So I was running CVG, then I found out, like probably everybody has, the G is insignificant and not necessary.
Silent.
Yeah, the G is silent. Yeah. I actually had a guy who was bashing drywall and putting it in his sub, and he kept getting trichoderma. And I was like, what are you using? And he had this like moldy piece of drywall that he was like bashing with a hammer, mixing it in his sub. And then you get the dudes who use the coir that has like trick already in it for the weed growers, and that stuff. I get these big 5 kilo bricks of like, it looks like, like they used to do weed, like brick weed. It's like a huge 5 kilo block. You can buy smaller blocks that are 400 and 800 grams. It's a big block, and I can make an enormous amount of sub with that. I think it's the same kind of thing they sell in the US. But Thailand. Yeah, there's lots of coke. It probably comes from Thailand. Lots of coconuts here, and it's cheap, man. I buy a 5 kilo, which is like, I don't know what, 12 and a half pounds, 0.2 pounds. It's like about $5, I think.
Wow. Yeah, it's cheaper than us.
Yeah, I feel sorry for you guys sometimes when I see the prices. I'm like, but your retail prices are higher too, so it kind of makes up for it.
And we have better Amazon selection too. So
yeah, that's
I sleep at night with the comfort of knowing my Prime is taking care of me. And then what really helps me sleep at night is knowing that Jeff Bezos just bought a 240-foot yacht.
Yeah, exactly. Yeah. Meanwhile, mine's on the back of some motorcycle taxis caught in the rain, but it eventually gets here. But yeah, just so now.
So you're just CV now.
Yep, CV, and I've been playing around with perlite because perlite is cheaper here.
Okay.
But it's kind of really hard. So when I do this is, again, I know, very unorthodox, but I have a giant like stock pot. It's something like it's they call them Indian pots here. It's like something you put a big curry or a stew in, and they're kind of flat. So it's probably a good 10 gallons, and I just dump my coir in there, and then I know this sounds really ghetto, but I just fill water in there. I just put it like a kiddie pool, so it's like a miniature kiddie pool. I've got my coir. And if I have vermiculite, I'll put it in there. If I don't, I just don't. Like, so half the time I'm running just straight up coir.
Yeah, people do that here too.
Yeah, I think I'm getting. I was on the vermiculite thing for a while, but occasionally you run out of vermiculite, and so I was just like, I'm just gonna run straight coir. The one thing with field capacity, I've noticed even here, being probably the country of origin, the consistency. Sometimes, it's all sort of pulverized a bit, macerated or whatever. But sometimes, man, you get down to the end of that block and it's almost like powder. So you've really got to work at the field capacity. So what I do is that's why I got such big forearms, not from something else, you guys. That's from squeezing coir.
Sure.
I basically I usually wear gloves because my hands are a mess. My skin is so dry, and you know. Anyway, I like my pretty fingernail. No, not really, but anyway, I wear gloves, so I just don't abrade the hell out of my hands, because if you're squeezing guar for like an hour, that stuff will rip up your skin. So I wear gloves, and I just squeeze it to field capacity, which you guys didn't watch the videos out there, and then I just like do it like everybody else does it. Like some of the videos I've seen, like on Yoshi, where I do it the same way. I don't use a hamburger press, but I do push it down pretty hard. And I've been noticing, I've been running some PE varieties or PE derivatives or whatever. And people say you got to press them down harder. And I've been doing that. But also because I'm running shoe boxes now, they're a little bit sturdier than I was running these sort of cat litter trays where I couldn't really press down very hard.
They give and.
Yeah, they get all flexible, and then when you move in the substrate. So I like that the shoe boxes are quite rigid, and I really sort of mash down the substrate. And my ratio is usually probably, I would say I would say almost 1-to-1 sometimes.
Oh, okay.
And that's because I'm running so many different crosses now, and I've got like a lot of cultivar cultigens that that I kind of circle through them, rotate through them.
Right.
So when I sub my plates, like if I get a full plate, after about 3 weeks of it, I try not to let it hit the edge, but once it gets close, I'll just make spawn. Right. So I'd rather have a backup bag of spawn sitting around for 2 months, and if something happens to the culture, I'll just pull a piece of grain.
A grain, I do that.
Yeah, single grain. So you don't have to continually sub your agar plates. If you've got a good bag of spawn laying on the shelf, if you lose I've lost GT so many times, it's freaking pathetic. Sure. But you just go back to that old bag of 2-month-old GT spawn, and you're like, I'm gonna pull a grain off there and pray, and there you go, start it again. So I usually run about 1 to 2, maybe twice as. I get confused where they put first the spawn, so it's like one.
The low number is always the spawn. Is kind of how
I do, like 1 to 2, maybe at a max 1 to 3.
Okay, that's about the same.
Yeah, it's somewhere around there, and it depends. I've
done
it both
ways.
If
you're running straight cocoa, I think 1-to-1 might be good, because the way I understand it, the whole point of the vermiculite is to hold on to some of that water a little bit longer, so that so it doesn't evaporate too quick. So if you are running straight cocoa, the evaporation process might be sped up a bit. You would want to rip through that cake, so you would want more spawn to sub, so that you don't ever dry it out or whatever.
With the crosses, I'm just trying to get F1 fruits so I can get spores.
Right.
So I'm literally with the shoe boxes now. I used to use these trays about the size of a cat litter tray, and they took maybe, I don't know, a week and a half to colonize, and then maybe another week and a half to fruit. And with the shoe box, they seem to just cycle through. I'd say maybe like you save 3 or 4 days. It's not.
Yeah, you save some time.
But because you still got to wait for all the, little metabolic stuff to happen. But I'm running shoe boxes now because of space. I don't have any space in my tents. So I'm trying to get those F1 fruits as quick as I can. And again, inspired by the master, these shoe boxes. I think if you're going to run a lot of cultigens, you're going to have to use shoe boxes. Yeah. You can't be running like a whole new cross in like a 66 liter quart mono tub. If it's crap,
that's the dead giveaway, right? Like, what you're doing this for. Do you have a wall full of shoe boxes, or do you have a wall full of 66 quarts? It becomes abundantly clear what we're in this for at that point.
I got a pile of those in my living room, like it's 20 high. It's like, why the hell did I buy all those? Like, I use them, but I don't know. It just becomes a certain point where, you don't have enough squirrels.
You don't. No, you really don't. It's true. So I got one little thing that, at least on this podcast, I don't want to get out of here without talking about, and that is, where is it here? It was something about children in China. What
Oh, I just pulled. Oh God,
This is guys. This is just my personal need. I probably could have asked him this privately, but he created. We're working on developing curricula for upcoming podcasts, and so there's the shared Word document, and he just had a note for the first podcast, and I never talked to him about it. So I got to hear this, dude. I got to hear this. I think it's a story.
I was getting grief for this last night from a girl who was I don't know, a little shocked at some of my life's choices. Long story short, I've been here for 18 years. About the fourth year I was here, my friend was going to China to teach English for a couple weeks.
Sure.
And he through this headhunter, this woman, they basically went there to teach English for a couple of weeks, and when I met her, she was very attractive, and she wanted babies, so I gave her babies.
Wow! So this was a Chinese headhunter who was headhunting more than just this head.
Yes, she was. Okay. She wanted a different head.
So if you're 23 and meing,
I'm not reproductively isolated yet. I'm not going to speciate. Well, I don't know. It could be it could be a new hybrid, a Chinese American, but God knows what I am. Americans are so mixed up. She might get like an albino. No, the kids are good. The boys like 13 now, and the girls like she'll be 11.
Nice.
So yeah, it's one of those weird things that I don't know. I know a lot of people that have kids, and I kind of, people kind of look down on me upon it but it was a very long story. If I ever meet any of you guys like later, it's a long story.
Cool. When we plan our Bangkok foraging ayahuasca retreat, we'll get the full story then. How about that?
Exactly. That's what we'll do. Some of you people out there, any yoga teachers or holistic breath work, or I got a list of things here on the wall somewhere. Yeah, if you guys know any of that, we'll set up we'll get all the hippies over here and start a commune. And then I'm
sure you can get some jungle square footage fairly inexpensively, right? Although I imagine the city is pricey.
Yeah, well, it's a fairly it's a very diverse country. They've got beaches and mountains and all kinds of stuff everywhere. Mm-hmm. It's a small country. I mean, like actual land area size, but it's really quite long and wide-ish. Mm-hmm. So yeah, there's all the good stuff. But yeah, if we'll hopefully maybe in the future we'll set up.
Yeah, that'd be fun. We'll do like Willie Myco style, and instead of Costa Rica, we'll just go to Bangkok, and I'm sure we can find all sorts of fun things to do in
there's like anything you want to do.
I'm sure. So I think somebody asked last podcast about psilocybe or actives in Thailand. Do you know of any? Have you found any? I know for a year or so you were out in the jungle looking for mushrooms, so I'm curious.
Never found a cube here. I did find some pan-cyans or tropicalis, whatever they are now.
Tropicalis?
Yeah, it could be some tropicalis. I'm not even sure. They were growing on dung, and I saw these little brown mushrooms growing on dung, and I found them one time, and I wasn't really sure, and there was only 4 or 5 of them, so I didn't bother. Mm-hmm. And then another time I went up to a guy's house in like what's called the Isan, which is kind of the farming area, Isan,
sure,
the northeast here, and they were growing. He had strawberries that he had mulched with buffalo cattle dung. Some kind of dung, some kind of ruminant dung, and they were growing all over in his strawberry patch. And of course, the local lady was like, "Oh, those are poisonous. Don't eat them. They're poisonous."
Right.
The time I found a huge field of cubensis in the Dominican Republic, and all the Catholic people around there were like, "Oh, don't eat them. They're poisonous."
Man, you telling me that story has me wondering if in general, right? Like those who are not lovers of mushrooms tend to have oh, don't touch that, honey, it might be poisonous. Don't you know? And then you got Alan Rockefeller going, you can lick poisonous mushrooms, it's okay, right? Like, definitely you can tell there's a difference in comfortability here. But you telling that story makes me think, I wonder if the whole origin story of like, don't eat it, it might be poisonous, isn't that there are a small percentage of very poisonous mushrooms, but more that just a bunch of fuddy-duddies that don't want you to partake?
Well, I would say in general, like, you shouldn't put unknown things in your mouth.
It's a good rule of thumb. I for some reason didn't even need anyone to tell me that. I have always lived by that motto. Yes.
I used to be in the poisoning hotline back in Tennessee. They had a number. Well, it was basically our lab number. And if you called there 24 hours a day, there was a good chance you'd catch one of the 4 or 5 people there, 6 in the morning or 12 at night. And you'd be surprised at how terrified a parent can, not surprised, but with good reason. They see their kid out in the grass, and next thing you know, they see a mushroom in their mouth. And they don't know if they've eaten it, if they've just chewed it, if they've spit it out, if they ate another mushroom. Strangely enough, you probably see all the dog poisonings. Dogs like to eat mushrooms. And so, in general, I don't put like random plants I see growing on the side of the road in my mouth. So I don't know why people. I'm also on the other side. I'm shocked at how freely people will like take random drugs that they buy off the internet. And, consume them. It's like,
and as someone who works in the ER, I can tell you that's a bad idea, because I take care of those people, and it's like, oh, you bought what? Well, the results are in, and you just OD'd on carfentanil, which is elephant tranquilizer, or the other one, the parents.
I got a pressure cooker I can sell you. Let me say. Perfect. You want to do drugs? I got a pressure cooker. I can
grow your own place. Grow your own. As a general rule, kids will not. If any people with young kids out there listening to this, as a general rule, if a little toddler tastes something and it tastes bad, they will not keep eating something that tastes terrible. So like, they might try chewing on an aspirin or on a Tylenol, but when they realize that it doesn't taste like a Tic Tac, they're not going to consume half the bottle. That's been my general experience. It has to taste good. Now, if the medicine has been made to taste good and they get a hold of a children's bottle, that's why a lot of those have the squeeze thing, so the syringe can go in there. And for those of you who are deaf, this probably looks bad. But it's not, it's very rare that we actually have true toxicology issues with ingestions from young kids. They just usually things that are bad for you taste bad, and you don't want to keep eating them.
Of course, the classic example is the amanitas, like the virus and the phalloides. They taste good, and they've got the delayed action and all that. But
that's the scary ones. Yes.
Yeah, those are the ones. If you are going to start learning field mycology, like field identification, the very, very first mushrooms you ever learn are the amanitas.
Yeah.
Those are the first ones, and they're a very, very beautiful mushroom. And if you do poison yourself with an amanita, you probably should stop mushroom hunting, because that would be like the people who over the age of 50 who can't like identify a Volkswagen bug.
Right. Like, it's a litmus test.
It's kind of like maybe you should eliminate yourself from the gene pool.
Yeah. Well, and they just might if given enough foraging opportunities. Yes. Yeah. Cool, man. We are at the 11 o'clock hour over here in Northeast Ohio. Your day is just about to begin. Again, I'm really praying one of these days the stars align, and maybe I need to start praying to God, and I'll get Alan back on here.
I'll get one of those masks next time, and I'll be like, "Oh yeah, Oakland. I was just I don't know how to imitate now.
I just got back from a foraging expedition.
I took a lot of photos. I did photo stacking.
Yeah, dude. That's what I want to talk to him about. Is the photo stacking, because the guy is like a wiki photography master. Like, even if you forget about his exhaustive knowledge of mushrooms and just nonstop foraging expeditions, he takes some really great photos. And when you're involved, my whole take is, in this like burgeoning interest in mycology and mushrooms in general that we're seeing, at least in the United States, I don't know if it's everywhere, having people taking really astounding photos, or shows like Fantastic Fungi. I mean, that stuff is really doing a huge service to the community by bringing, more interest to it, because it's showing off what we all already know, is that these mushrooms are fascinating and they're worth paying attention to and learning about and all that kind of stuff. So I'm hoping to learn some of the tips and tricks, and hopefully we'll talk about microscopy with him a little bit, and DNA sequencing. And we're going to definitely talk about both those things with you a little bit more as time goes on. Guys, if you have not liked, subscribed, commented, all that usual stuff, please do. It just helps the channel. And I definitely am doing this so more people can connect with more of this information that's happening on the show. So I appreciate that. And then guys, Ed's got a channel. It's in the last, it's in his last podcast that was originally Alan's podcast, and it'll be again in this one that was originally Alan's podcast. So check it out. He's got all these great, like, pretty short videos that when you're done, you'll oftentimes know something you didn't know before, and you'll pick up some tips and tricks, and seems to be doing really well. So anyway, Ed, thank you for again waking up and helping me out. I really appreciate it. No problem. And we'll talk again soon.
Okay, man. I'll see you guys later. Thanks for listening.
I did. Okay, guys. Next week we got Slightly Feral, super unorthodox, out of the box, really fascinating guy. I hope you guys can find the time Monday to check it out. If not, watch it at your leisure. Anyway, till next week.
These socks are still hurting my ears.
Well, hold on, dude. I don't think I'm done yet. The glitch. I don't think it.
It's okay. I got pants on.
I know. Great.
I got pants on today, you guys. Don't worry.
Yeah, dude. This thing is not shutting off.
Mine's acting weird too.
I don't know what to do.
Before the next person, you might want to make sure to test it out. Mine's acting funny too. It looks,
dude, if I lose this whole broadcast, I will be so fucking mad.
It's not. Would it not be?
Yeah, it's just so like all I can think to do at this point is because I tried to leave the studio, it wouldn't leave the studio. I tried to end the broadcast, it won't end the broadcast. I think maybe I'm gonna try to just reload it. All right. Anyway, let's see what happens.
Good luck. I don't know if you can hear me. I just see me now. Oh, my socks look funny. I gotta get something.
All right, dude, you're still live. I hope you didn't pick your nose or anything.
No, I didn't. He's still alive. No, probably
whole thing. And when I went back into the studio, it said still live. So, okay, I think I'm going to get to end it now. Anyway, guys, thank you very much. Wow, tonight was just all sorts of unexpected pleasures. Hope you guys enjoyed Ed. Until next week. We'll do a GoFundMe for some headphones for Ed.